Hello, everyone. So, this is the second talk in the ecological dynamics series. This is the second talk in the ecological dynamics series. And today we have Arshed Nabeel, who's a PhD scholar in Vishu Gurur's lab in Center for Ecological Sciences at IIC. So, he'll be talking about this cool new paper that they have put out, which is mainly on guppy dynamics. So, hang around and let's enjoy the talk. Thanks. On to you, Arshed.
Uh, Thank you, Milan, and thanks everyone. Thank you, Fishers, for inviting me to this talk. So today I will talk about some thoughts on how do we model systems in biology and ecology. Uh, so before we start, if whenever you see this green triangle in your screen, it is a slightly more detailed mathematical details of some of the aspects of the talk which are strictly speaking not necessary for the flow of the talk. So I might rush through some of the details whenever you have the slides. So even if you do not grasp all of it, or if you sort of ignore some of these details, you will still get the gist of the talk. With that, let's start. So, while modeling systems in ecology and biology, we are often faced with this question of what is a good model. Uh, Specifically, like how do we choose a model, and what is the level of complexity that we need to incorporate to get a right level of description of the model? So, I will talk about I will broadly visit this question from the context of a specific case study that we did recently about population dynamics of Trinidadian guppies. So this will, in equal parts, be a technical talk and also a bit of a perspective talk. So as a prelude, I'm sure all of you know this, the logistic model for population dynamics, where you model the dynamics of population of any species as a very simple differential equation which broadly represents the following fact, which is the population grows with some intrinsic growth rate R to a carrying capacity K. It's an extremely simple model, and it's almost a toy model. This is ecology 101. And you treat your entire population as one unit, and you only keep track of the population size at any time. You do not think about, for example, age structures of different traits the organisms might have or whatever. And it's based on one simple assumption, which is density dependent population growth. So let's think about this question. Can a highly simplified, almost cartoonish model like the logistic model be actually useful in modeling population dynamics of actual real world populations? Let's think about this question specifically in the context of Trinidadian guppies. So this data set comes from a long-term eco-evolutionary study in several streams in Trinidad, where they monitored the population dynamics of guppies in these streams. So we'll ask the question, what is the good model for their population dynamics? As you can already think, this it's not a simple organism or a simple system, right? It has a very complex population structure and life history. It has sexual reproduction, there is age structure and all those things. And there are also several biotic and abiotic interactions. There's predation, competition, and other abiotic pressures and whatnot. There is also eco-evolutionary dynamics. In fact, the study was established to study eco-evolutionary dynamics, and there is environmental effects from seasonal changes so uh, at first sight, this surely you would not expect a population like this to be describable by simple models, right? So let us uh, start by keeping our biases aside and start to look at what the data tells us. So we'll use this broad approach that I've been using in also in my PhD research called data-driven modeling or data-driven model discovery. which is it's a suite of techniques that helps you recover dynamical models directly from empirical time series data. So the important fact to note here is that the specific model and the structure of the model are usually inferred from the data and not specified based on first principles or hypothesis by the model. So in this specific context, uh, our input will be a time series, here being the population of guppies, sampled at some sampling interval. Here, the sampling interval is every month from some underlying continuous time generating process, except T, which is unknown. We only have these samples of the population. Our goal for now will be to discover a model that describes this dynamics. We will choose the framework of stochastic differential equations that describes the dynamics of your underlying process X. A brief intro to SDs. A stochastic differential equation will have two parts, which describes the dynamics. A deterministic part, which sort of governs, which sort of describes your average dynamics, plus a stochastic part, which will tell you about the fluctuations about this average dynamics. Specifically, the stochastic part has this g of x term, right? Which means that the strength of your fluctuations are dependent on your current value of your state variable. In this case, you're the population size. So this is like called a multiplicative or state dependent noise. And when noise is state dependent, it can actually make qualitative changes to the actual dynamics of the system. So how does the technique work? Briefly, it's a two-step process. First, we will estimate the deterministic and stochastic part with what is called Kramer-Moyal averaging. Basically, you estimate your finite time, the forward differences, just as you would do in any numerical analysis, uh, and sort of take a conditional average. I'll get back to the conditional averaging bit in a moment. Once you have estimated the deterministic part, you can estimate the residual fluctuations in your forward differences that are not explained by this deterministic dynamics, this average dynamics. And then you take the variance of this residual fluctuations. That gives you, again, a conditional average variance that gives you the stochastic dynamic. Broadly speaking, or somewhat loosely, but you can think of the deterministic part as the mean of your finite time derivative, delta x by delta t, and the stochastic part as its variance. Now comes the question, how do there was an angle bracket, the conditional averaging that I had written down in the previous slides, which did a bit of heavy lifting. Now we'll sort of think about how do we actually do this conditional averaging. The simplest way, which has been standard in literature for decades, which is to sort of divide your state space into small bins and take average in each bins of this, the fluctuations. But this an even better way, which we recently introduced, and many other people have also uh, concurrently arrived at similar solutions, which is to use sparse regression to fit your F and G as uh, using a candidate basis of terms. But a common basis would be a polynomial basis. It's not just a polynomial regression, though. What we will do is a sparse regression using a specific technique called STLSQ, but you can also use other techniques. The reason to do this is that even if your library or your basis set is very large, you will try to fit your functions with as few terms from this library as needed. So this allows you to discover simple parsimonies and interpretable models that describe the dynamics of your state variable X. Coming back to our system of guppy populations. So we have a time series data from these streams of guppy populations. And we have this tool, which we have also built Python package which does this. So we'll use this data-driven ST discovery technique to actually get an interpretable model of population dynamics. And what do we get? Perhaps the title of the talk gives it away, but we find that the population dynamics of these Trinidadian guppies are, in all streams that we measured, are very well described by a stochastic logistic model. So this is a stochastic differential equation whose deterministic part is simply your logistic growth function. Importantly, there is no higher order effects like alley effects. The stochastic part says that your fluctuations, your noise fluctuations are actually proportional to n. This is called environmental stochasticity in literature as opposed to, say, demographic stochasticity, where your fluctuations will be proportional to square root of n. So now, okay, so I just told you that I used this technique and the technique told me that this is the model. How do we know this is the correct model or at least a good enough model for the population dynamics? So we need to actually validate this model against the data. Uh, to do that, we will make predictions about certain statistics, specifically about the steady state distribution and the autocorrelation function of the system or of your populations. And here, what you see in orange curves are what the model predicts. And the blue curves and the blue histograms are what the data says. So as you can see, the model is a fairly good match for the underlying dynamics. Uh, so now we have this model and we know that it's a reasonably good model. What do we do with it next? You can sort of do a lot of analysis and make predictions about using this model about the fluctuations, how the fluctuations affect the population dynamics, how resilient is the population to fluctuations and other effects. I'll highlight a couple of things that we did in this paper. One is like we said that the fluctuations are environmental fluctuations which scale as n. Can we say something more about what is the source of the fluctuation? To do this, we use certain techniques from signal processing literature. Basically, you linearize the system around its fixed point. Think of it as a linear system. So think of the fluctuations as input signals coming into the system and the observed population sizes as the output signals from the system. And then you can actually you can write down this simple relation between the power spectrum of the driving fluctuations and the observed population dynamics using the transfer function of the system, so-called system. And what do we see? Once we reconstruct the input fluctuations using this technique, we see a peak at around 11 to 12 months, which is exactly the annual seasonal cycles. So we see that the fluctuations in the system are mostly dominated by seasonal fluctuations. And we make some sort of hypothesis about the strength of these fluctuations, the impact of these fluctuations on the population and the relation to the thickness of the canopy, for example. Next, you can also make predictions about the resilience of the population. We can specifically ask which of these, when we have measured multiple streams, we can ask which of these populations are close to a population collapse and which are reasonably healthy populations. To do this, we will characterize the effect of the fluctuation strength on the population resilience. Specifically, we had this stochastic differential equation model, the stochastic logistic model. You can derive the steady state distribution as a gamma distribution of this following form. And then you can track how the mode and the mean of the distribution changes as you increase your strength of fluctuations. This is a plot that shows the same thing. So basically, since this is a fairly dense plot, let me slowly walk you through this. The black curve here shows you the mode of your population distribution or the most probable state for the population for a given value of fluctuation strength. And the blue dots are the average value that you will observe. Uh, this was plotted based on simulations. And the basically the beta, the green region is where the most probable state or the mode of the distribution is above zero. Once you get out of the green region, extinction state or population size equal to zero becomes the most probable state. If you look at the vertical purple line, that is the estimated beta for this particular stream. I think that this stream was called Taylor. And uh, based on this, that the estimated beta for the stream is quite far from the point of collapse, which suggests that this stream is reasonably healthy. As opposed to another stream, Upper Lalaha, which where your beta is actually quite close to collapse. So we think that the population might be at a greater risk of collapse due to environmental fluctuations in this particular stream. Note that what we quantify here is the risk of collapse due to strength of fluctuation, due to stochastic environmental fluctuations alone. If you just, as you can see, we are only varying the strength of fluctuations and not your, not for example, the growth rate or carrying capacity, which means that your traditional early warning signals to detect collapse will not actually detect this risk of collapse. So the SLM is actually telling you something more that your traditional models and techniques will not be able to tell you. Right. Uh, so I think that brings me to the end of whatever I wanted to speak today. I think I took far less time than I was anticipating to. But let me summarize whatever I was saying so far. Uh, Basically, we saw that the population dynamics of guppies in these streams were very well approximated by a very simple model, namely the stochastic logistic model, which basically captures a logistic growth for the population as the deterministic dynamics and environmental fluctuations at the stochastic dynamics. These stochastic logistic models were also reasonably shown to be good models for microbial population dynamics, which is also an interesting study because these are microbial populations which are interacting within a large community. So, in a sense, the community interactions all average out to sort of to the form of an environmental stochasticity when you are tracking the population of only one population. Even though this is a highly simplified model, the model offers a good fit to data and it also allows us to make important predictions, specifically predictions about the nature of fluctuations, the resilience of the populations, risk of collapse, etc. Uh, does this mean that, okay, the stochastic logistic model is all we need to describe this population dynamics of guppies? Of course not. Depending on the questions we ask and the precision and the details of the things we want to know, more complicated models will be appropriate. For example, if you are going to study the eco-evolutionary dynamics, uh, like a population dynamics that uh, completely ignores any sort of evolutionary dynamics will obviously be inadequate. Uh, but the point I'm making here is that uh, even simple models can tell you some things. and some really good insights despite the system being very complex. And then I also want to sort of put down this idea of top down data driven modeling as a fairly powerful approach to build models for complex real world systems in biology and ecology. Uh, so, indeed, these systems have been designed to handle complex data indeed, my own research is on using these sort of data-driven top-down models to come up with unifying models for the collective movement of a wide range of species of fish. Finally, let me close on this sort of broad uh, sentence, which is that very simple models can be surprisingly powerful even in modeling fairly complex seeming systems. So most of this talk was based on two of these papers. The guppy population dynamics is recently out as a preprint. Uh, it's called Noise and Determinism in Trinidadian Guppy Population Dynamics. And the technique itself, the technique to derive dynamic equations from data, it was published last year in AMNAT, and it's also available as a Python package. Let me thank Vishu, my mentor, for all the support and also for the freedom to sort of step out and do parallel things during my PhD. And also to Harman, who sort of robbed me into this project and led this project, and for giving me this chance to work on this very interesting system. And let me also thank all the collaborators and co-authors of the various manuscripts, and also Fishers for organizing this and inviting me. Let me end there, and uh, thanks for listening, and I'll open the floor for questions.